Assalamualaikum warahmatullahi wabarakatuh, everyone. So this is a pre-recorded lecture for chapter 4 titled Techniques in DNA Technology. Okay, in this chapter, focus on introduction to DNA technology. So in a modern DNA technology, okay, we have to know there is a production of recombinant DNA. Okay, recombinant DNA, and then in this recombinant DNA, production of recombinant DNA involved with DNA extraction, electrophoresis, polymerase reaction, production of recombinant DNA, and then the DNA sequencing. So what is recombinant DNA? Recombinant DNA is the where the molecules of DNA from two different species, okay, that being inserted inside the host to produce a combination of DNA that will be give benefits to science, agriculture, okay, pharmaceuticals, okay, and also farm animals, okay. So that is the importance of recombinant DNA. So the process of production of recombinant DNA involve several steps that I've been mentioned just now. Okay, start from 4.2 until 4.6. Okay, then in this chapter 4, okay, we will focus on the application of the recombinant DNA technology. So last few weeks, okay, we already covered on the DNA extraction, okay, DNA amplification, electrophoresis, and in this week, okay, we'll continue on the recombinant DNA, production of recombinant DNA and DNA cloning. Okay, so common methods, okay, to clone species of DNA is using bacteria. So the common bacteria apply is the E. coli, and inside the E. coli they got a plasmid that will become the vector. So the vector is the site, okay, where the gene of interest, okay, will be combined, okay, and then after the combination, then okay, the recombinant DNA will be inserted inside the host. Okay, so this is the plasmid, the round structure that is isolated from the bacteria E. coli. Firstly, okay, the isolated, okay, the bed of the plasmid then will be inserted with the gene of interest. Okay, this resulting as a recombinant DNA molecule. Okay, so recombinant molecule is again is the combination of molecules of DNA from two different species. First is the species with the gene of interest. Another one is the DNA, okay, from the bacteria. Okay, so the plasmid then will be inserted, okay, into a host. Okay, the plasmid then will be inserted inside the host to produce clones of identical cells. So then the dividing bacteria will replicate, okay, the recombinant plasmid and pass it to their descendant. Okay, so the foreign gene is cloned at the same time. So here class, okay, this is the plasmid, okay, of bacteria. So there's a restriction site here. Restriction site here means that there is a cutting site lah, okay, where the plasmid will be cut, okay, using restriction enzymes. Okay, so the enzyme will be applied to cut at the restriction site of the plasmid. Okay, and then after cutting the plasmid, it will produce sticky ends. Okay, melekat lah. So lekatan tu the melekit. Okay, and then the sticky end then will be, okay, added, will be combined with the gene of interest that being extracted from plant or animals. Okay, so what is gene of interest? Okay, gene of interest is a gene or molecules of DNA, okay, that will express, okay, proteins or that will give benefits to a human. Okay, and this gene of interest can be have from plants or animals. Okay, so to have this gene of interest, you must cut the DNA, okay, from plant or animals, okay, using the same restriction enzyme that you used to cut the plasmid just now. Okay, as you can see, number one, they have the recession enzyme to cut the plasmid. Okay, and then the same recession enzyme, you cut the plant and animal with the gene of interest. So they will give the same end. Okay, and then because of the same end, this then allowed, okay, the gene of interest to bind at the sticky end of the plasmid. So this, there will be one possible combination. Okay, after that, after combination of the molecules of DNA, okay, by the gene of interest and the plasmid, okay, this then will involve with DNA ligase, okay, to seal the strands, and then this the end product is the recombinant DNA molecule. Okay, so again from this slide, you must know it involved with two types of enzymes. First is the restriction enzymes, second is the DNA ligase. Okay, and the restriction enzyme, okay, must be the types of restriction enzyme, okay, must be the same you use to cut the plasmid and also the DNA with the gene of interest. Okay, then the DNA ligase function is to seal the strands between the plasmid and also the gene of interest. Okay, so for cutting and joining DNA, for this technique, two types of enzyme, restriction enzyme called restriction endonucleases. Okay, this act as a biological scissor to cut the DNA at specific sites. Okay, the DNA here referring to the DNA from the plant or animals with gene of interest, and also DNA from the plasmid. Okay, then DNA ligase to join, okay, the two strands. Restriction enzyme ORE recognize specific sequences within the DNA molecules and cut at the sugar phosphate backbone. And DNA ligase will join the DNA fragment that have complementary sticky ends or the blunt ends. So restriction enzyme or restriction endonucleases act as a biological scissors that cut, okay, the plasmid, okay, and of the bacteria. Another one is to cut the DNA from plant or animal with the that give gene of interest. So the type of the RE, okay, must be the same. See, so that they will have produced the same sticky, the same end that will allow them to combine, okay, between the DNA, okay, and the DNA ligase used to seal the two DNA molecules or two DNA strands. Okay, after you produce the recombinant DNA just now, okay, recombinant DNA then will be cloned, will be amplified, will be multiplied the numbers. So the function of cloning will produce mass number of this cell of a recombinant DNA is to make many copies of a particular gene. Okay, so you will have numbers of same you will have the gene of interest in a large amount. Okay, so and then to produce a protein product. So there are the two functions of cloning here lah. Okay, so an example of genetic engineering from this picture, as you can see here, this is the gene of interest. You bind with the bacteria or a plasmid of the bacteria, produce the recombinant DNA. The recombinant DNA will be inserted inside the host, okay, inside the bacteria again, and then it will, the clone of the bacteria then will amplify, or bacteria will multiply, produce clone of bacteria, say with the specific gene. Okay, researchers can isolate, okay, the clone gene from bacteria, okay, to for use in basic research or to endow an organism with a new metabolic capability. For example, they could produce plant, okay, with pest resistance. Okay, that is the function of the amplification or the cloning of the bacteria. Next is that it can help in producing protein with medical uses such as human growth hormone, insulin, okay, and then in a large quantities, okay, by cloning the bacteria cultures, okay, in large numbers. Okay, As you can see here, okay, the combination of the DNA and then help in production of insulin. So from this picture here, okay, this is the bacteria, and inside the bacteria they have the bacterial chromosome and also the plasmid. So the plasmid will be extracted out from the bacteria, okay, and then will be cut using the resection endonucleases, and then the resection endonucleases will also be used, okay, to cut the gene of interest from the cell. This is animal cell, and then ligase will be added, okay, to seal the combination of the DNAs, okay, then produce recombinant DNA. Recombinant DNA then will be added inside the host, okay. So the host here is the bacteria. Then the bacteria will clone itself, okay, amplified, multiplied. So not amplified, it will be multiplied, okay. Then this and then inside the bacteria, okay, the gene will express, okay, producing protein. So the protein could be harvested from the clone cells, okay. So then the clone cells, so then the protein, okay, from the clone cells, okay, can be used for basic research, okay. For example, for the protein could be the human growth hormone, helps in stunted growth, and also to produce to help in blood clots in heart attack therapy. Other than harvesting the protein from the express gene from the host cells, okay, the function of the recombinant DNA, okay, also help in producing in multiplying the numbers of gene, okay, that will use in basic research. For example, the gene can be applied, the gene of interest then can help in give resistancy for the plants toward pests, and also help in the cleaning of water by toxic waste, okay, seawater from toxic waste, okay. So there are two basic, okay, importance, okay, of the cloning, okay, of the recombinant bacteria, okay, from the host cell. First, okay, by having the protein expressed by the gene of interest. Second is amplifying the number of gene of interest that can be used in basic research on gene, okay, basic research on gene. So this is a picture, okay, of bacteria, okay, with the plasmid extracted out. So number one is the plasmid, okay, and this is the human cell with the gene of interest, black color. So using the same restriction endonucleases, okay, number two will be the restriction endonucleases to cut the plasmid, okay, producing sticky ends, okay, and then the same restriction endonucleases will use to cut the gene of interest in human cell, okay. Then both DNA will be combined, okay, and then the DNA ligase is number 3, okay, to seal the combination of DNA molecules from both species, okay. And then number 4 is the recombinant DNA, okay. Then the recombinant DNA will be inserted into back to the bacteria, or we call as the host cell, okay. Next, after producing the recombinant DNA, okay, this then followed with the sequencing. So the ultimate goal is sequencing is to determine the complete nucleotide sequence of each chromosome. Sequencing also known as mapping a genome. So we have the gene, okay, that been multiply, okay, inside the bacteria, right. So kita dah tahu dia dah amplify, dah banyak, producing banyak DNA. So we want to know the sequence of the DNA that yang berjaya di amplify, yang dia berjaya di multiply. So you nak tahulah sequence dia, adakah lari ke, apakah new gene yang terhasil, kita tahu kan. So we want to know by sequencing to map the genome. So that is called as sequencing, to mapping the genome, to know the bases, to know the sequence of the new recombinant DNA. So in this sequencing step, okay, we must have DNA fragment up to approximately 800 base pairs. Dia tak boleh banyak sangat, tak boleh banyak sangat. Approximately about 800 base pairs in length. Okay, so the DNA will be cut, okay, and the DNA fragment then will be cut, okay, into DNA will be cut into fragments, and then okay, sequencing will be implement. Okay, so sequencing technique, okay, here applied, often called the dideoxyribonucleotide chain termination method. Okay, this that is developed by a British scientist called as Frederick Sanger. Okay, so from the term is that from the term itself, dideoxyribonucleotide chain termination. Okay, so means that it use them, it needs, okay, involved with the termination of the involved with the dideoxyribonucleotide that will terminate the chain during sequencing. So what is dideoxyribonucleotide? Okay, the dideoxyribonucleotide is a nucleotide bases, okay, yang mana dia punya apa, color, okay, fluorescence, okay, added, and then with the presence of this dideoxyribonucleotide, dia akan break the DNA cloning producing fragments. Okay, so dia akan terminate the chain. Okay, it's okay. Let's look what is dideoxyribonucleotide. Okay, before this kita tahu kita ada ribonucleotide, kita tahu kita ada deoxyribonucleotide, but this is the first time for you to hear the terms of dideoxyribonucleotide. Okay, so this dideoxyribonucleotide only be mentioned during the DNA sequencing. Okay, so technique in sequencing, okay, it involved with, okay, technique in this in sequencing, the fragment of DNA, okay, to be sequenced is denatured into single strands and incubated in a test tube with the necessary ingredient for DNA synthesis. So basically DNA sequencing ni you kena synthesis your DNA, macam DNA replicates lah. Okay, then in DNA synthesis, okay, involved with a primer, okay, number 2 DNA polymerase, involved with deoxyribonucleotide, and then the dideoxyribonucleotide. What is dideoxyribonucleotide? It's a deoxyribonucleotide that tag with a specific fluorescent molecule. What is fluorescent molecule? Molecules that give presence of colors. Mana nanti help in detection during UV light, nanti detect lah. So kita tahu tu base apa. Kita tahu dia A ke, adenine ke, guanine, tyrosine of apa? Apa lagi? Pyronine. Kan? So kita tahu the basis, okay, because of the specific fluorescence. Okay, so this is also basis yang mana color, itu saja. Dia basis tapi bercolor, that being apa, color, that being tagged with color. Okay, so that is dideoxyribonucleotide. So all this, okay, the deoxyribonucleotide, the dideoxyribonucleotide, DNA polymerase, and primer must be added inside the test tube because kita nak amplify the DNA, okay, the strand of the DNA during sequencing. Tak apa, later kita akan tengok video. Menda akan bagi video so that you can understand well, okay, on the DNA sequencing, the steps involved. Okay, so synthesis of each new strand start at the 3' end of the primer. Okay, you letaklah semua primer contain, and then after letakkan primer, the process of synthesis, then dia after letak primer at the 3' end, then dia akan start dengan the deoxyribonucleotide. So then after synthesis of each new strand start at the 3' and continues until a dideoxyribonucleotide is inserted. Okay, so bila adanya dideoxyribonucleotide, this then will stop. It will terminate, okay, the chain. Okay, sebab tu kita panggil dideoxyribonucleotide chain termination. Okay, first primer. Okay, let me briefly explain about the DNA sequencing. The DNA sequencing is that, okay, first is the strand of DNA. Okay, kita akan break the DNA, and then after the DNA break, okay, we have one strand, and each strand tu dia akan bind, one strand tu dia akan bind with a primer to start the DNA replication. Okay, after primer tu dah bergerak, after the DNA polymerase catalyze, okay, function of DNA polymerase to catalyze the addition of bases to the template strand tadi kan. So DNA pun bases pun akan being complementary, bind with the bases, dia akan produce a new strand of DNA. Okay, then after that, okay, dideoxyribonucleotide, okay, the DD bases ni yang tag with the specific fluorescent ni, dia akan bind at the end of the strand. Okay, bila dia bind je dekat strand, okay, dia akan bagi stop the process of the elongation of the strand. Okay, so eventually a set of labeled strands of various length is generated. So dia akan basically the dideoxyribonucleotide, dia akan bind daripada bases of the template DNA at different length. Nanti kita akan tengok macam mana. So because of the different length of strands that akan produce, this help in understand the sequence of the template. Okay, nanti kita akan tengok video. So this is ribonucleotide that usually found in DNA, and this is the deoxyribonucleotide, okay, in DNA. Ni RNA, ni DNA, and then the dideoxyribonucleotide is the chain terminating inhibitors. Okay, dia mana dia akan stop, okay, the elongation of the new DNA by DNA polymerase. This the dideoxyribonucleotide used in the Sanger method for DNA sequencing. Okay, then after they dah stop the fragments from elongate, okay, dia akan produce fragments with different sizes. Okay, so the mixture then will be separated by passage through a polyacrylamide gel in capillary tube with shorter strands. Okay, will move faster to the negative end. Okay, dia punya teori same with the gel electrophoresis. Okay, mana the shorter fragments you akan move faster to the negative, okay, poles. So the fluorescent detector then senses the color of each tag at the strands, and strands differing by as little as 1 nucleotide in length can be distinguished. So then because of they are different in one basis je, so dia boleh detect lah dia adalah bacaan sequence untuk that DNA template. Okay, so results, the color of the fluorescent tag on each strand indicates the identity of nucleotide at its end. So using the fluorescent tag from the dideoxyribonucleotide tadi help untuk kita tahu ni nucleotide dia, basis dia apa. Okay, so which is complementary to template strand, then can be read from the bottom on top. Okay, so let's see the machine here. So this is a template strand, the green color. Okay, inside the test tube letaklah reaction of mixture, you letak primer kan, you letak DNA polymerase to catalyze, okay, the addition of bases to the DNA template kan, and then you guna DNTP, then juga you add with the dideoxyribonucleotide, okay, the DDNTP. Okay, and then as you can see here, okay, now green is the template, okay, of the recombinant DNA tadi yang you dah buat. Nak tahu kan dia punya DNA punya sequence kan, so you cut the DNA template, kita akan dapat. So then you will have your primer, and then primer dia akan bind to the template. Okay, After that, number two, primer elongation. So dia akan start dengan elongate of the DNA template. Okay, so this is the primer. Start with primer. Primer elongation is that the DNA polymerase will catalyze the elongation of the primer. Okay, start from the 3'. So kita akan letaklah bases yang complementary dengan template yang green ni. Okay, kita akan letaklah bases yang complementary dengan DNA from the green template. Okay, dah complementary. Kemudian the dideoxyribonucleotide yang tag with colors, dia akan bind, okay, randomly. Okay, randomly dia akan bind at the end of the elongation. Okay, so bila dah bind je dekat at the end of the elongation, then automatically the elongation will stop. It will terminate. Okay, bila terminate, dia akan bebas lah. Then another primer akan bind balik kepada the template. Okay, so this process continue until complete lah. Okay, then akan dapat fragments of DNA. Okay, with primers tadi yang dah elongate at different length. Okay, paling panjang dan paling pendek. Okay, dia akan run inside electrophoresis. Okay, gel electrophoresis, capillary gel electrophoresis. Number 3, then dia akan separate the DNA fragments. Okay, yang paling ringan akan berada paling awal gerak. Dia akan baca paling ringan dan paling berat. Okay, akhir sekali paling berat. So ada laser and detector to identify the bases. So bases apa yang di-identify? The dideoxyribonucleotide tadi lah yang tag with fluorescence. Okay, yang akhir sekali. For example, paling berat tadi blue, paling ringan tadi warna pink. Okay, so dia from there dia boleh tahulah ni bases apa. For example, blue tadi bases dia C, cytosine. Okay, and then after paling panjang, kurang panjang sikit, warna purple, so that is tyrosine. Okay, and then followed with green, adenine. Okay, so then dia boleh tahu the sequences of the recombinant DNA using chromatograph. Laser detection of fluorochromes and computational sequence analysis, kita pun dapatlah the sequence of DNA. This is example of printout from a DNA sequencer. A for adenine, thymine, guanine and cytosine. So recall what we learned today is that in DNA sequencing, okay, it involved with the DNA template, okay, primer, DNA polymerase, the deoxyribonucleotide, and the tag deoxyribonucleotide called as dideoxyribonucleotide. So the tag deoxyribonucleotide ataupun dideoxyribonucleotide ini, okay, dia tag dengan color fluorescence that will help in identify the bases. Okay, dan dia hanya bind to the template. Okay, bind complementary to the template at the elongation primer pada at the end of the elongation. Okay, bila dia bind je, dia akan terus terminate. So, sebab tu kita panggil dia hanya ada pada at the end of the elongation because fungsi dia adalah bind and then terminate. Okay, so here because of the termination, okay, bind and terminate. Okay, so nanti kita akan dapat numbers of fragments of DNA at different length. Okay, and then run, okay, dekat gel electrophoresis. Okay, paling ringan akan bergerak paling pantas, okay, to the negative pole. Paling berat lambat lah. Okay, dan dia akan detect ikut urutan, di mana beza dia adalah satu bases to one fragment tu dia akan beza from one another fragment melalui satu bases saja. So dia akan bantu untuk kita identify the sequence of DNA, okay, by bases lah. Okay, and then laser detect, okay, and computational sequence analysis. So this is automated DNA sequencer. So last is the application of the recombinant DNA technology. Okay, what is the purpose of having this technology? For medical application, for pharmaceutical products, environmental application and agricultural application. Okay, this is application of the recombinant DNA for gene therapy and also for basic research on protein. In medical application, in gene therapy is that, okay, for example here, this is okay, what is gene therapy? Gene therapy is you use the vector, okay, the retroviral as a vector, and then you insert gene of interest or the normal gene, okay, into a patient yang mana dia ada sakit, masalah gene, dia ada masalah dengan dia punya gene, for example cancer, okay, ataupun ada penyakit yang berkaitan dengan gene kan. Okay, mana the cell from kita akan beri, okay, kepada pesakit, okay, gene yang mana ada gene yang baik, yang sihat kepada pesakit supaya dia akan menghasilkan gene yang baik. Okay, that is called as gene therapy. Okay, a retrovirus has been rendered harmless. Okay, the retrovirus yang harmless will be used as a vector, okay, in this procedure, okay, which exploit the ability of retrovirus to insert a DNA transcript of its RNA genome into the chromosomal DNA of its host cell. Okay, again, this is a picture of a retrovirus, okay, and this retrovirus dia ada DNA, betul? Not DNA. Virus have RNA. So this is RNA of the virus. Inside the RNA of the virus, kita akan produce recombinant DNA. Kita akan insert with the normal gene, okay, untuk normal gene yang sihat untuk manusia. For example, to treat the brain cancer, okay. So the normal brain cells yang mana untuk treat the brain cancer tadi, to stop dia punya multiplication of the cancer cell of the brain, will be inserted into the viral DNA, RNA, so dia akan produce a recombinant. So this recombinant of the viral RNA tadi, okay, dah bind dengan normal allele untuk patient tadi. Kita akan letak ke dalam bone marrow, okay, of patient, okay. Kita akan letak dalam the retrovirus inside the bone marrow cells of patient yang sakit gene tadi, genetics dia, genetic disease. Okay, and then after that, the normal tadi dia akan bind with the chromosome, okay, of the cells, bone marrow cells. Then dia akan clone, hasilkan banyak, multiply, okay, then injected into the bone marrow, okay, of the patient. Okay, so that kita dengan intention is that this is as a therapy, okay, so that dia akan hasilkan sel yang baru yang akan memberi genetik yang baik, okay, kepada pesakit tadi supaya it will treat the genetic disease. Okay, so this is gene therapy. Other than gene therapy that is for medical purpose, for pharmaceutical product is that it help in production of human insulin. Okay, the recombinant DNA, okay, from pancreas cells, okay, di mana from pancreas cell tadi dia ada insulin producing gene. Dia akan extract this gene of interest, okay, bind with the plasmid from bacteria producing recombinant DNA, inserted inside the host, okay, bacteria. The bacteria tadi akan clone, dia akan multiply, dia akan amplify. So then each bacteria cell tadi, it will express protein, okay, the gene tu akan melalui proses of expression, okay, producing protein or known as insulin here. And then people or doctor or pharmacist they will extract, okay, the scientist will extract and purify the protein tadi, and then dia akan jadikan that as a human insulin. Understand? Okay, so basically the human insulin tadi akan jadi banyak lah, okay, instead of sedikit dia akan dapatkan banyak human insulin because of the cloning process. Okay, next is the other than insulin, hepatitis B vaccine. Okay, So in hepatitis B vaccine, this vaccine is produced by the hepatitis B virus. Okay, kita ambil virus tu, okay, jadikan dia sebagai vaksin. Mana kita ambil the virus, the antigen of producing gene, okay, of hepatitis B kita akan extract from the virus, yang mana memang that gene hanya untuk producing hepatitis B kan. So kita ambil that gene of interest, kita bind with the plasmid, okay, of the bacteria. The recombinant DNA will be inserted inside a new host called as yeast cell. The yeast cell akan multiply, clone, dia akan clone banyak. So dia akan undergo process of expression producing hepatitis B antigen. So this hepatitis B antigen then will be extract, purified, dan dia akan membentuk hepatitis B vaccine. Kita akan cocok dekat orang supaya, okay, dia dah introduce with this virus, so dia akan produce their own antibody lah. Human tu yang telah diintroduce dengan this vaccine, dia akan dapat antibody. So body kita protected lah sebab kita ada antibody untuk melawan the hepatitis B virus. So next, other than for pharmaceutical and medical application, for environmental application is that, okay, the recombinant DNA help in biodegradation of oil spill, okay, tumpah minyak dekat laut kan. So because of the tumpahan minyak, because of kapal-kapal kan, kita hasilkan bakteria yang boleh hidup dengan minyak. So kita akan tabur the bakteria. So bakteria bukan bakteria biasa-biasa, bakteria known as recombinant bacteria. So this recombinant bacteria, okay, they can eat the oil that spills on the sea. Okay, so using genetic engineering, okay, they have microorganisms yang mana can grow rapidly on oil. So the microbes, they akan eat the oil spill, okay, by breaking the oil into non-toxic and non-polluting materials. Thereby this end up cleaning up the site. Okay, some of the oil utilizing microbe can also produce surface active compounds that can emulsify oil in water and thereby removing the oil. Okay, that is the function of the recombinant DNA, okay, in environment. Next, for agriculture application, transgenic animals. Okay, for animal farming, okay, they apply this recombinant DNA for production of better sheep, better fish, better chickens, better cow, better goat, sheep and pig. Okay, in sheep, okay, they produce better sheep with better wool, better fish with mature in shorter amount of time, better chicken with more resistant to infection, better cow that will increase the milk supply and meat with lack of fats, leaner meat, and better goat, sheep and pig to produce human protein in their milk. So these transgenic animals, okay, the is called as animal farming. Okay, animal farming. Other than animal farming, okay, mana dia ada produce animal with better protein, better wool, everything. Okay, the transgenic animals also, okay, using the recombinant DNA, okay, also help in producing farm animals. Okay, what is farm animals? Farm animals means that is the short terms for pharmaceutical animals. Mean that animal tu dia boleh menghasilkan ubat. Okay, kita hasilkan transgenic animals that give pharmaceutical values. Okay, this transgenic sheep, for example, they carry a gene for a human blood protein. Mana this gene dia boleh hasilkan protein yang bagus dalam susu dia. Okay, di mana dalam susu lembu ni, okay, dia akan hasilkan protein, dia ada protein that help in inhibits, okay, dia boleh merencah enzim that can cause lung damage in patients. Okay, okay, so patient ni dia ada sakit cystic fibrosis. Okay, di mana this cystic fibrosis ni that biasanya infect the saluran pernafasan manusia lah. Okay, yang akan cause production of thick phlegm or thick fluids kan, okay, dekat bahagian saluran pernafasan. So by having this protein from the farm animals, okay, dia akan inhibit, okay, the enzyme yang akan menyebabkan penyakit itu. Sebab tu kita panggil farm animals, give pharmaceutical value based on dia punya production lah. For example here, susu dia ada protein yang mana boleh inhibit enzim, boleh menyebabkan penyakit cystic fibrosis dan juga chronic respiratory diseases. So easily purified from the sheep milks, the protein then is currently under evaluation as a treatment for cystic fibrosis. Other than for transgenic animals, also have transgenic plants. So this is agrobacterium tumefaciens, okay, di mana the DNA from the plasmid from the bacteria, okay, dia akan bind dengan gene of interest, okay, menghasilkan recombinant DNA. Plasmid with the gene of interest. To cut them, kita guna the same restriction enzyme. So then for production of transgenic plant, okay, the recombinant DNA dia akan masuk balik ke dalam host. So the host then akan bind dengan DNA of the plant. Dia akan masukkan dalam plant culture lah, biasanya plant cell culture. Okay, dia akan bind dengan plant cell culture of the plant cell and also the bacteria, the host. Okay, so then because of the mixture of the cell culture, plant cell culture with the bacteria, okay, dia akan allow the process of transformation di mana the plasmid tadi, the recombinant DNA tadi, dia akan move to the plant cells. Then the new gene, dia akan bind with the DNA of the plant. Okay, then the plant will undergo cell division. Dia akan produce DNA with the gene of interest. Okay, so what is the importance of the transgenic plant? Okay, dia akan hasilkan the transgenic plant give characteristics that like it will give herbicide resistant, pest resistant. Okay, that's all. Okay, for the importance of the application of the recombinant DNA. So today, okay, we already covered on chapter 4. Okay, we learn about the recombinant DNA, which is the modern DNA technology. Okay, how to produce the recombinant DNA, what are the enzymes involved. Okay, and then we learn about the DNA sequencing, how to sequence how the DNA sequence is produced. The important terms that you have to know in DNA sequencing is that there is a di-deoxyribonucleotide that is tagged with fluorescent color. Okay, mana dia akan bind pada template of DNA, bind pada DNA at the end of the fragments. Okay, then after dia bind je dekat at the end fragment, dia akan terminate lah, dia akan terus tertanggal, so akan produce, and then at the end of the sequence kita akan nampak different size of fragments. Okay, after DNA sequencing, okay, kita belajar tentang the application of DNA recombinant terhadap medical, agriculture, okay, dan juga agriculture ni termasuklah plant dan juga animal, producing transgenic plant and also transgenic animals. Okay, I will give you the link for the DNA sequencing so that you can understand the flow, okay, in the production of so it will helps you in understanding on how to read the sequence of recombinant DNA DNA lah. Okay, so now is the recombinant DNA, so kita akan tahu the sequence, or we will understand the sequence of DNA, okay, by the application of DNA sequencing. Okay, alright, thank you. Assalamualaikum.